In this video, we are going to discuss the structure and functions of transferrin. So what is a transferrin? Transferrin, the transport of iron. So it is a protein which is going to transport iron. There are many examples, many types. In blood plasma, we call it as serum transferrin. In egg white, we call it as ova transferrin. In milk, we call it as a lactoferrin. And these transferrin, if the iron is not present, we call it as apotransferrin. They are a very important antibacterial agents because they deprive microbes of their iron. So iron will be removed from the microbes. The transferrin are also present in our ears, serving to clean the eyes after some irritation. The structure of a transferrin. See, the transferrin, it is a glycoprotein. It is having a single polypeptide. There are, we divide it into N-terminal lobe and C-terminal lobe. And each terminal is again subdivided into subdomain. In N-terminal, we call it as N1, N2 subdomain. In C-terminal lobe, we call it as C1, C2 subdomain. And these subdomains will form a cleft. Okay, they form a cleft. And that cleft, the iron 3 will be binded. So that is the binding site for the Fe3. And we can see here from the structure, we can see we have got a iron. This is the 2 subdomain, and we have got iron. Similarly, where here also we have 2 subdomain, and we have got an iron present here. Now we have to know about the coordination. So in coordination, this iron Fe3 plus is having a distorted octahedral geometry, and it is coordinated by carboxylate oxygen, 2 phenolate, that is from tyrosine, and an imidazole from histine amino acid. And additionally, we have exogenous, a carbonate, which does not belong to this protein chain. Separately, we have it. It is very important. It is called a synergistic ligand. For binding of this Fe3 plus, carbonate is very, very important. So this is how the structure looks like. The coordinations are seen here. Now we will see how the binding of iron takes place. See, the Fe3 plus, once it comes inside the protein, there will be a conformational change in the subdomain so that they make some space so that iron can come and sit inside. So some kind of a conformational changes will occur. And when it occurs, when this iron comes, it alone is not important. Along with it, the bicarbonate or carbonate should also be present, which is shown from this reaction. That is apotransferrin, transferrin without an iron. And iron is going to come to it. A bicarbonate is there, and we form a transferrin. That is transferrin with iron 3 and carbonate, and H plus has gone. Since H plus is gone here, we can understand that this particular transferrin binding and release, it all depends upon the pH values. So synergism between carbonate and Fe3 is due to the simultaneous binding to each other as well as to the protein. Not only this carbonate anion can be used, but other anion can also be used. For example, oxalate, salicylate, malonate, citrate, etc. And we can see why Fe2, from this anionic ligand set, we can see that Fe3 binds more strongly than the Fe2. And this transferrin, using this transferrin, not only Fe3, also gallium 3 plus and aluminum 3 plus can be used for transportation among the cells. Now, how does this occur? So Fe2, first it gets oxidized to ferroxidase, Fe3, then it combines with carbonate and goes to the apotransferrin, and they form a transferrin. Now the transferrin is there, transferrin is ready with iron. And the cell which is in need of iron, it will produce a transferrin receptor. It will have a transferrin receptor in the plasma membrane. It will send a signal that it requires an iron. Now, to that particular cell, to particular this cell, the transferrin will be carried by the blood, and thus it will come to the cell. And in that cell, the transferrin receptor in the plasma membrane and the transferrin will bind together, and they form a complex. Now, this complex is still outside the plasma membrane. It has to go inside. Now, how it occurs? The cell does it by a process called as endocytosis. It engulfs the whole receptor inside it, and it forms a vesicle. Now, this vesicle will break down, and the iron will be released if the pH is lower. For example, for serum transferrin, if the pH is 5, the ion will be released. For lactoferrin, the ion will be released if the pH is 2 to 3. Now, once the ion is released inside the cell, the transfer receptor goes back to the plasma membrane by a process called as exocytosis. Thank you.